Thank you everyone for attending this very first webinar for the Advancing Treatment Award. We're now going to hear from our Director of Research, Jessica Lee.
Thank you, Giacomo, and thank you everybody for joining this webinar where we're going to talk to you in a bit more detail about our 2024 Advancing Treatments Award. We're going to be joined by 3 members of our Research Review Committee. So that includes Ang Armstrong, a person who's affected by MND, has been affected by MND. We have our Chair of the Research Review Committee, Richard Mead, and our Vice Chair, Jill Richardson. I'm going to kick off though by providing a short presentation about the Advancing Treatments Award. I'm just going to kick off with an introduction to My Name's Doddy Foundation. So for those of you who don't know, we are a UK MND charity. We were established in 2017, so around about 6 years ago, by ex-professional Scottish rugby player Doddy Weir. Doddy, if you didn't know him, was a larger-than-life personality. He was diagnosed with MND in 2016, and when he was diagnosed, he was shocked and surprised at the lack of treatment options. So he set out on a mission to raise both awareness of MND and funds for MND research. He established the foundation with a vision of a world free of MND. We are primarily a research charity. We're focusing on translational research to accelerate the development of new treatments. And so far, we've already committed over £11 million to MND research, with an additional 6 million and more to be committed in 2024. Last year, we published our first research strategy, Catalysing a Cure, which outlines our strategic priorities. They include research to validate therapeutic targets, to accelerate new treatment, and to improve translation. So a key way for us to implement that research strategy is through our Advancing Treatments Award. So here our aim is to invest in high quality research to develop a pipeline of new treatment options. In 2023, we invested £1.27 million across 3 research projects through this scheme. This year, we have slightly more funding on offer, 1.75 million. So the scope of the awards is around target validation and preclinical studies. We won't accept applications around target identification. We do currently have another award open at the moment, the Discovery Network. There's accepting applications in that space. And we also don't fund clinical studies through this scheme. So focusing on these 2 areas that we do fund, the kind of projects that we're looking to support for target validation are studies in vitro and in vivo models to demonstrate that modulating a specific target can result in pathway modification and rescue of disease phenotype. We're also looking at studies that can define downstream biomarkers, whether that's target engagement or pharmacodynamic biomarkers that can be used in preclinical studies going forward. For preclinical studies, we're looking to support studies to demonstrate drug efficacy, safety, to determine human dosage, and to identify clinical strategy. So that could be looking at patient stratification approaches for a given therapeutic. I'd like to mention the guiding principles for drug discovery and development in ALS. So these are principles that were published last year by a working group of academics and industry experts that outline best practice for drug discovery in MND. Quite a few of the questions that we'll ask throughout our application process are linked to this, and it's things like, why did you choose your selected models? That's something that we want you to think about really, really carefully during your application process. And some questions to ensure that we're developing really robust preclinical strategies and projects. So for example, can you demonstrate target engagement in vivo? So later on, when we talk to Jill and Richard, we'll talk in more detail about these guiding principles. But if you haven't already looked at that document, it is available on our website, and I'd encourage you to do so. So the application process for this award. So we announced the award in February. Our expressions of interest close midday on the 17th of May. Please remember that they do close at midday because that caught people out last time. We don't want that to happen again. We will invite probably a maximum of 10 expression of interest applications to the full application stage. We expect to do that over the summer, so either June or early July at the latest, and full applications will be due in September. We hope to provide a relatively rapid funding decision in October, and we're hoping that projects will launch in January 2025. I do want to point out that contracting has been taking quite a while for some of our projects. So if you are successful in reaching full application stage, we'll ask you to share our grant agreement with your universities as early as you possibly can, just so that we can work through any problematic clauses so that we can get started with research as quickly as possible. So thinking about the assessment criteria, so what are we looking for in these awards? Well, one thing that we're looking for is really good lay summary and appropriateness of PPI activities. For the lay summary this year, when you submit your expression of interest application, if your lay summary is not thought to actually be appropriate for a lay audience, we'll return your application straight back to you to redo the lay summary before it actually moves on to that internal triage stage. So it's something that's really important. We've linked some guidance onto our website and within the application form for support in developing a lay summary. We're looking for projects that have really high potential to impact people living with MND. We're looking for projects that are scientifically robust and show a level of scientific innovation. We're looking for strong justification for the use of animal models. So that's the kind of the type of model that you've chosen, but also the number of animals that you want to use and how you want to use them. We will assess the suitability of applicants, so making sure that that team is really well-rounded, it's got all the expertise and knowledge within it to conduct the proposed research. We're looking for projects that have that potential to rapidly translate towards the clinic. So we want you to have thought about your commercialization plan. So what IP, what are the considerations around IP that need to be made? What considerations need to be made to ensure that you've got the right partnerships to quickly take forward that research? And we want to see that you're thinking about what is going to happen at the end of your grant with us to make sure that the research is taken forward rapidly. We'll ask you in your application about risks for the project. We want to see that you've identified the relevant risks for the project, but that you also have appropriate mitigation strategies in place to address them if those risks do materialise. And we want to see that you have a cost-effective and realistic budget. So that's the assessment criteria. I've spoken to you a bit about the scope of the awards. You can obviously go and see much more detail on our website. And if you have any further questions, please just get in touch with us. So that was the initial presentation that I wanted to provide. And I'm now going to introduce Ang Armstrong, who is a member of our Research Review Committee. Hi, Ang.
Hello there.
Hello. Can I just start by asking you, why did you get involved with the foundation, and why do you think research into new treatments for MND is so important?
Well, the obvious reason I got involved with the foundation is due to my being affected by MND with my husband, who was diagnosed with MND and had a very, very short life with it, really. He was diagnosed in August 2021 and passed away in November 22. And during that time, we realised that obviously there is no treatment options, and it is just managing the symptoms. And it just felt so unacceptable for something like that to be, for any condition that is life-limiting like that, to have nothing to turn to. So yeah, as a family, we've definitely got behind the foundation to support all of the work they're doing.
And what does this research mean to you? So, you know, the idea of supporting new treatments for MND, how important is it to you individually and from your perspective of sitting as a lay representative on our research review committee as well?
It's really important for people to have options in the future. I know it was too late for us. Again, going back to the fact that it just seems, you know, so unreal that there isn't anything when you look at the treatments and how cancer now is being treated, and you know the 1 in 3 people will get cancer, but 85% of them will live past 5 years. And I'd love to see that being available to someone with MND to at least have a chance at life, basically.
Exactly. And you've been involved with our research review committee since its inception, which isn't long ago. So we've been running for a year now. We've gone through 2 grant schemes already, one of which was the first year of our Advancing Treatments Award. So we've obviously seen our kind of first tranche of applications come through, and we've taken some learnings on, you know, what makes a really good application and what makes not so good an application. So I just wondered if you could share some thoughts with us about, you know, what you think makes a good and a poor application.
Of course. Well, as a complete lay person, my background is marketing and recruitment and HR. So seeing some of the lay summaries in the initial phase that came through, I had to do a lot of Googling. So in order to understand what the concept was, what was being proposed, and that for me took obviously longer than someone with a scientific background, but I was prepared to put the time in to do it on this occasion, but knowing there were going to be learnings from this so that we could take it forward. I know that you can't necessarily do these lay summaries without particularly technical information. A globule is a globule or something. You can't call it something different, but it was much more tricky for me and more time consuming to really assess what the proposition was so that I could really get my head around it. So for me, I would have liked to have been able to read it and see more bullet points and things like that, so, and keep sentences shorter and use less acronyms. There seem to be a world of acronyms in the scientific world. And you know, obviously you can spell it out the first time and then acronym it, but even for me, I like to think of, in my world, in the commercial world of let's just say Eloqua and Salesforce.com. I mean, you may have heard of some of those platforms, but the way they talk to each other, the integration and the API, and so I may be using language now that is maybe some people do know what those things are as well. But in my world, talking to my colleagues, that would be fine. But if I was talking to a client about what I was trying to do for them, it would be much more simplified and it would be bulleted. And I guess I was thinking it potentially as if you're talking to a child. It's stripping it right back. And what are the key points really? What's the salient point? What is it you're trying? So I always think of the principle of a press release when you should be thinking of who, what, why, when, where. So if you can use those things, who am I? What am I trying to do? Why am I trying to do it? And just as short sentences or bullet points. And for the lay summary, for me, it's just that all I need to see is what are you trying to propose here? How long is it going to take? And what's the outcome, essentially?
Exactly. And the lay summary is just so important. On our research review committee, we have 5 people who are affected by MND. So every application is reviewed by at least 2 people who have been affected by MND. Plus our panel meeting is a mix of people affected by MND and scientific reviewers. So for us, making sure that we have a good lay summary is so important, not just for the review process, but also for how we communicate the project afterwards. So we want to do, we want it on our website and we want to communicate with our supporters. So yeah, I completely agree that getting a lay summary is something that maybe is overlooked. The importance could be overlooked by potential applicants.
Yes, I think so, Jess, because I do feel sometimes if something's overly complicated, it's a bit like doing your expenses, isn't it? Sometimes you just have to put your mind to it to get to do it. It's those tasks that you do that you just procrastinate on them. And if something's too tricky, or just feels like it's a big job, you always leave it, and it never really. But I mean, I guess it's like picking up a book that excites you. You read that bit on the back, and that's what makes you want to turn the pages.
Yeah, exactly. And I guess, is there anything else that you learned from our previous round about what makes a good versus a poor application?
Too much detail is not always needed necessarily, because I do think it's about being succinct. Across the entire application, sometimes there's a lot of repetitiveness. You read something in the lay summary, it felt like it was a bit of cut and paste into the body of the main applications, which, sometimes we'd all, if something's technical or it's giving us information, we would rather read 3 pages of really, really good content than 27 pages of repetitive sentences.
Yeah, exactly. I think that's something that came up as well, is how it's ordered in the application. So the projects that had, subheadings and bullet points, the committee felt it was much easier to get through those applications and, find what they needed from them.
Yeah, and sometimes less is more. I mean, going back to my other job is, I interview Apex and Visual Force technical architects, and the CVs are just jam-packed with, and it's really important. All of these skills and qualifications they've got are really important, but sometimes that's what the interview's for. If you give everyone everything, I mean, I know this is a different situation, but in that initial application, it's about having that taster. So long as you give enough information that enables a conversation or enables you to get through to the next stage, for me, that's what's really important, is to keep it really succinct. I think it's a really good way that Jess highlighted here, a particular lay summary, and some of you will be able to read this and go, oh yeah, I know exactly what that means. So there you go. This is fairly common for the type of lay summary, which is, as you can see, fairly condensed text, and there's quite a lot of information in there that for someone like me, I would need to Google, and I'm just trying to, I mean, some of it you can work out, and you can see there that there's where I had to Google some of those things, and then the bit in the middle, some of that could be bulleted, but it is just technical information. And is it really adding to what the lay summary is trying to express? And as you can see there, what is the most important bit? If you can think about when you're putting your summaries together, the who, what, why, when, where, what am I trying to achieve? What's the scenario? What am I trying to achieve, and what's the outcome?
And is there anything else that you think people who are affected by MND, who are sitting on our panels, are particularly looking for in applications when they do come to us?
Yeah, I mean, I know that when I was looking at them, I was looking at the potential outcomes, really. How is this going to impact people in a positive way who are, suffering with MND? I hate to use the word suffering, who are living with MND. And I'd be looking at how it's administered potentially in the long run. I'd be looking to see whether any contraindications have been thought about, because sometimes the solution feels simple, but actually, what if somebody's got an allergy? It's all those, because you all think, I would be looking at it from my own personal experience, as with the other people who've been affected by MND, but I'd be looking for how soon it can happen as well. How speedily can we put this out there? That would be another key thing for me, because I think time is of the essence now.
Exactly. And I think that's something that runs through everything that we've said in our research strategy. We want to make sure that we're accelerating research. So we want our applicants to really think clearly about how that research can move to the next stage as quickly as possible. And I think outlining it not just from a scientific perspective, but from a layman's perspective as well. So for people who don't, know the drug discovery process in great detail, knowing that the applicant has thought about the different things that is going to help on that commercialization pathway is going to be really useful for our lay members.
Totally agree. I just feel that if the I's haven't been dotted and T's crossed of the next stage, sometimes it's okay to identify something, but where's that going to go next? If there's another, I know that this is something that you've talked about before, Jess, but for me, if this funding that's needed to get to this stage needs twice that to get to the stage after, how can we change? How can we look at that in a different way? And I'd also, some of the work I've done when I've been reviewing some of the applications is to try to understand that it's not already being done, or it's not something that somebody else is already looking at, and that maybe there's a collaboration, but is it best use of funds is another thing.
Yeah, exactly. And it's that's right. So best use of funds, not just from, is it being done elsewhere, but is how the funding is proposed for the project. Is it being used appropriately? So, is the methodology being done as succinctly as possible so that you can reach your next inflection stage with, enough funding, but with, the right funding in place. We don't want to see, too many parallel work packages that actually aren't needed, and therefore funding going into projects that might not be essential. So I think it's thinking about how we can streamline both the methodology to ensure that we've got cost effectiveness, but also avoid that duplication of effort that you're talking about.
Yeah, and if there is a project that shows a eureka moment that other people can then go on to use as a basis for new research, because there's so little known really still, what is the cause, and getting treatment options is one thing, but sometimes it's like there's so many different things that can be looked at for this particular disease as well.
Yeah, exactly. And you also mentioned collaborations and partnerships. And for this award round, although lead applicants have to be based in the UK, we're actually opening the round internationally. So applicants can be based anywhere else. And obviously only academics can actually receive grant funding, so only universities, but we're happy for industry to be collaborators on projects, and actually industry could help that rapid translation towards the clinic as well. So we are interested in these multidisciplinary, multi-institutional teams to take this research forward for sure.
Yeah, bring all those minds together.
Exactly. So before we finish this session, is there anything else that you wanted to raise and I guess give, a few final thoughts on what we want to see in applications from a person affected by MND's perspective?
I guess really just to summarise some of the points that I've already made is short, sharp in delivery. Think about the who, what, why, when, where. It just seems works in PR in a PR way. Bullet points, highlighting any particular words in bold. Use acronyms, obviously, because there's many of them, and just try not to make it overly technical, and keep it bulleted, short sentences, short and sweet.
Yeah, that's perfect. Thank you so much, Ange. That's really helpful, and we'll see you again in the Q&A session in a while.
Okay.
So we're going to now bring on the chair of our committee, Richard Mead, who's a senior lecturer in translational neuroscience at the University of Sheffield, and our vice chair, Jill Richardson, who's the head of UK neuroscience and executive director at MSD. Hi, Richard. Hi, Jill. Thanks for joining us.
Hi, Jess. Hi, everyone. It's a pleasure. Thank you for the introduction.
Thank you. So you've both been involved in our research review committee for just over a year now. Like Ange, we've gone through 2 award rounds, so we've got some learnings about what works and what doesn't. So can I just ask you both, I guess, what are your learnings from our previous application? What works well and what doesn't work so well? Maybe Richard will kick off with yourself.
Yeah, so I think you've done a really nice job of explaining the value and the need and for a good quality lay summary. So I won't labour that point anymore. But we've seen successful applications that have gone from kind of standard drug repurposing approaches through to quite novel strategies that could have actual quite broad applications across motor neuron disease and potentially in other areas as well. But in general, we're looking for strong background rationale, So that could be, you know, genetic, epidemiological, preclinical data, and the supporting rationale that this is a good target and approach in motor neuron disease. And ideally, I think that has potential to be applicable in sporadic motor neuron disease, I think is the most desirable types of target. I think we like to see, obviously, with all of these types of applications, some good quality underpinning data that tells us why you're applying, why now. You know, so as an applicant, what's your unique angle and why have you come to the point of wanting to take this therapeutic forward, which normally entails some kind of data that you've generated in your own model systems or with your own therapeutic approaches that is kind of highly relevant. I think one key thing for me is a good understanding of, and we'll come on to the kind of guiding principles which talk a lot about this, a good understanding of kind of basic pharmacology or drug discovery, really. You know, how do you go from kind of one stage to the next? How do you identify the relevant doses in your in vivo systems, let's say, based on your in vitro data? What does that mean in terms of clinical translation? What's the line of sight to a kind of clinical therapeutic? And can you give good evidence that you can find a dosing regime or an approach that will work, will engage the target in humans and have the potential to modify the disease, but also have you considered the potential for toxicities and side effects, and that those will be minimal and that the risk benefit will be very much in favor of a kind of therapeutic approach? You talked about risk, Jess, in your presentation as well, and for me that's really important. I think quite often when we as academics particularly put grant applications in, we try and downplay the risks or suggest alternative strategies. Whereas I think for drug discovery, we recognise it's a very risky business. And there's lots of projects fail, and that's the expectation, actually. And we don't want to stop people from taking risks, but we want them to demonstrate that they understand what those risks are and have a good quality risk mitigation strategy, which might be an alternative approach, but it also might mean that you've got a milestone in there, which means that we will stop at a particular stage if we've not been able to achieve a particular outcome. I think the other thing, just to finish off with, is the co-development. There's a lot of discussion in the guiding principles about target engagement biomarkers, and those are really important. We can go into that in more detail, but I think knowing what they are, or maybe co-developing your biomarkers that you're going to use to assess efficacy across your preclinical model systems, but also with a sight into clinical testing as well, is quite important. So those are the kind of, for me, just thinking about that question, some of the key things that spring to mind.
That's really useful. Thanks, Richard. And Jill, your thoughts?
Yeah, thank you, Jessica. It's a pleasure to be here as well. And just to clarify, MSD stands for Merck Sharp Dome, in case people don't know what the acronym is, which is the same company as Merck in the US, right? And we are very active in neuroscience research, and I actually lead a program in the motor neuron disease area. And so for me, I suppose the attraction of this particular board was around the translational focus. And yeah, ultimately, and it's important to hear from the people affected by the disease, because, you know, there is obviously a place for basic research, but the involvement of those people that directly are affected really puts the focus on, well, where's this going to lead? How is it going to translate into a drug one day? So, you know, I've been on many different boards, MRC, Wellcome Trust, and so on. And sometimes when I bring up the questions that we've just talked about, you might get an academic on the panel saying, well, that's not for us, right? We don't need to worry about that. So I think the real difference here is that actually we do need to worry about that in terms of these grant applications. And so some of the points Richard's already raised, you know, about line of sight to the clinic, about innovation, about, you know, even thinking about safety, and working in a pharmaceutical company, you know, all of these questions are looked at early. You can't always know, obviously, you know, just from a basic analysis of whether a target's going to be safe. You know, you really have to do the appropriate tolerability studies, but, you know, there are some targets that you just know you wouldn't touch because, you know, the knockout mouse, they might all drop dead or something, and, you know, that wouldn't be your first choice of target. But of course, there are always caveats as well. But, you know, the other thing in thinking of how you're going to move the research forward is thinking about, in terms of the target rationale, the emphasis on human biology and genetics, and, you know, I take the point about relevance to sporadic disease as well, but I think for some of the broad mechanisms, you know, if you have got some link to genetics as well, I think that's very informative, and human biology, expression data, and so on. And with the animal models, there's always a lot of, you know, debate about animal models for all of these neuroscience disease areas. But I think for ALS, you know, some models might be fit for purpose for some mechanisms, but you can also use other models that demonstrate that mechanism is disrupted and that you have a way to modulate that. And then if you've got the human biology to complement that, then I think it's less of an issue to have a bespoke ALS model. Line of sight to the clinics are thinking about the PD biomarkers, the clinical biomarkers, what's known in the literature about, you know, how you can measure these, because ultimately, once you get to that point of taking a target all the way through, you've got to have all those plans in place to know that you've really tested the mechanism, tested at the right dose, and you know, many times when we see clinical trials that have failed, you know, people walk away from the targets, and it may be that it wasn't tested properly, right, and it hasn't gone up to the highest level of target engagement. So I think to have some handle on that, on what that level is preclinically, is really, really important. So I'll stop there because I think some of the other responses I might give will be in more direct to some of the questions you have, Jessica.
Yeah, I think that's really useful overview from both of you. And there's probably a few different pieces that I want to pick up. I guess maybe if we go first to the two different scopes that we're looking at. So first is target validation. So for those target validation studies, I guess thinking about the targets that are being proposed to validate, we want them to demonstrate relevance, safety. We want to see their differentiation from other targets. How would you advise applicants to go about being able to show that in their application? Are there specific things that you're looking for in applications?
Do you want to start, Richard? Or?
Yeah, I think it just comes back to the question of, because obviously we're talking about preclinical here, but you may have, so whatever, are you talking about in the grant itself, how you build that target validation package?
Exactly,
Yeah.
exactly.
Yeah, so obviously there's basic stuff like it's there, it's expressed in the right cells at the right time, that kind of guilt by association, and then you can move on to, you know, if there isn't already existing genetic evidence, is there a case that you might be able to build some genetic evidence around the particular target? So there are huge resources now and potential collaborators in those types of areas. When you look at things like Project Mine, the whole genome sequencing data that's available, and looking not just at coding but also at non-coding changes, looking at associations with not just say incidence but also things like survival. So there are lots of ways of potentially bolstering the genetic evidence, let's say, around a particular target. I think you need to think about then the in vitro models and the in vivo models, and that they are, and I think this is where, I guess, over the last 10, 15, 20 years, you know, the kind of use of humanized in vitro model systems has been quite powerful, that you have a potential to use a kind of humanized system to look at validating your therapeutic hypothesis in those types of model systems. And then from an in vivo perspective, and I think the guiding principles talk about this, and Jill's already mentioned, it might not be that you need a particular ALS mouse model, although there may be some that are very, you know, that's my area of interest and expertise, and there are some I think that are very relevant for particular mechanistic hypotheses. If you don't have that, it's not the end of the world. You may be able to build a validation case around your in vitro data with some wild type in vivo data showing that you can get reasonable target engagement in the CNS with your approach. So it's really about thinking about all the different lines of evidence that you can bring to bear. So anything that's missing, you really need to be thinking about that package and whether there's anything that's missing that you can potentially strengthen your case by adding that particular component in.
And that's another thing that comes out through the guiding principles, isn't it? So generating multiple different lines of evidence, so that kind of independent replication, whether it's in different labs or using different models to build up a hypothesis. Sorry, Jill.
So actually, I'll pick up on the point you just said. So many times we've seen a paper where, you know, a molecule might work in a disease model, and then, you know, it's just one publication, and then you're aware of many other labs that can't replicate the data, right? But of course, that doesn't get published, and you know, that's a big issue. So you just learn about this data through informal sort of communication and so on. And we've certainly replicated or not replicated some of the studies in that respect. So if we start a new drug discovery program and there's literature, you know, we'll go and take a tool molecule and test it to replicate that before just investing a huge amount of money and just taking it for granted that what's out there is correct. So, you know, obviously academic labs can't necessarily have the resources to do that. But the point is that, you know, if there's more data from more labs to support the target, then, you know, that gives you a lot more confidence. I suppose, as well as, I mean, for us, it's emphasis on the human biology. So coming back to the animal models, you know, yes, I think sometimes it gives you a false sense of security that it reverses something in an ALS model. But for me, say like a mechanism like oxidative stress, you know, there's literature coming out, there's data from you know, competitor companies showing that oxidative stress biomarkers seem to correlate with disease progression, for instance. So if you had a model that exhibited oxidative stress, and you can show that the level of target engagement you need for your particular drug reduces that oxidative stress, and that, you know, then correlates with an effect on function in the model, then that goes a long way towards supporting the case for that particular target, but not in the absence of other data, right? So you still need that human expression data, as Richard alluded to, and, you know, whether you've got iPSC, some of the complex models. You know, I think if I was trying to convince a clinician to take, you know, a program forward to patients, I would heavily weight the iPSC data, I think, you know, human data. And if I saw, although it's hard to generate those complex models, I think you know, the reward of having that data is huge, right? And it really helps to make the case. The other thing, I think, when you are, you know, there's obviously gene-based validation, tool-based validation. I think over the years, I've seen, you know, a lot of grants where people use tool molecules at such high concentrations, and it's just not really very relevant, because, you know, up to 10 micromolar, for instance, and you see an effect, and then it's claimed that it's working through this mechanism, but actually, you know, the EC50 or IC50 in the primary assay is nanomolar, right? So, you know, whenever we see that from a drug discovery point of view, we immediately have, you know, a lot of concern about whether the effect is real. So it's important to use different tool molecules, and ideally, you know, show with knockouts as well, if it's an agonist, that the effect is lost in the knockout and this sort of thing. So again, I appreciate it's not always easy for academics to get hold of the right molecules or the resources, but certainly in the collaborations we have with academics, we really, really push that, and we do help them. So, you know, you can always ask companies to provide those molecules under a CDA or MTA or whatever. So, you know, don't just necessarily take commercially available tools, you know, reach out to companies, and I'm sure that most will oblige with providing those molecules.
That's great, thank you. And I guess the second scope is around preclinical studies. So we will support anything from efficacy testing, safety testing, all the way to determining the right dose for human studies, and thinking about that clinical strategy. So I guess just thinking about, maybe this is one for Richard to start with, demonstrating efficacy in preclinical models. You've already mentioned the importance of target engagement studies. But the other kind of things that we want to see is that we can modulate that pathway in either in vitro or in vivo, and that we can start to see those functional outcomes in the model. Is there anything else particularly that you're looking for there? What kind of pieces of, I guess, experimental methodology are you looking to see?
So I think, I mean, one thing to think about when you're using a preclinical animal model in particular is it's not just about the model, but what you're also evaluating is the molecule and the ability for the molecule to access the target tissue, the target cell. So that's called a kind of pharmacokinetic, pharmacodynamic relationship. So pharmacokinetics, what the body does to the drug, so where it goes, how it gets metabolized, and pharmacodynamics, what the drug does to the body. So you really want to be able, and that's quite often that's missing. So as Jill's kind of intimated, people will use a tool molecule at a concentration that's doesn't really fully validate your particular target, you will also find that people will use doses in preclinical animal models that don't really engage the target in the tissue, and so don't provide good evidence for your particular biological pathway. So things like understanding that you need to build a knowledge or an understanding of how dose relates to exposure, relates to target engagement, relates to a pharmacodynamic effect on the biological pathway, and then ultimately can modulate the really complex physiology of the human motor, or in the case of an animal model, animal motor system, to actually slow down degeneration of that system. That's the kind of evidence base that you're trying to build up. So I agree completely with Jill, and we you know, very much value the use of in vitro model systems and humanized systems, really powerful. But quite often it's the animal model systems that will give you the kind of that interaction of complex physiology with pharmacokinetics, with disease, degenerative phenotypes, that allows you to really start to build a strong case for a particular therapeutic approach. And obviously, when you're thinking about translating doses to human, as soon as you start to get that data from, say, a mouse model or a more complex animal model, you can then start to think about, okay, what could my doses in patients be, in humans be? And you can, even though you're not potentially as part of this project going to do that full piece of work, you need to be able to show an understanding that that's kind of where we're going. And with this data, we'll be able to use that data to generate a prediction of a dose in human at some point in the future. Yeah, I think that's...
Yeah, so it's having that pathway in mind, isn't it? So, you know, what is your commercial plan to get to the clinic? What are the different pieces of data that you need, and how are you going to get there? And I guess the other thing that we want you to think about, as well as the kind of scientific components, is, you know, thinking about any IP issues that might come up, any partnerships and collaborations you need to rapidly translate towards the clinic. And I guess, Jill, we've spoken about, you know, the importance of IP protection in translational research. I don't know if there's anything that you want to say on that.
Yeah, and obviously that's a primary consideration, you know, for a pharmaceutical company, that you've got a long period of exclusivity. And you know, particularly for CNS drugs, right? Because they're very expensive, very long, but you know, you have to often do imaging, so they're really big investments. So you've got to have a return on that. You know, it's a commercial business at the end of the day. But I suppose sometimes, and Richard brought this up in terms of repurposing. So sometimes I've been on grant reviews or boards, and you know, it's talked about repurposing, but if there's a very limited IP protection, say by the time you get to actually launching the drug, you've got 1 or 2 years, you know, it's very difficult, because, you know, whilst there may be sort of phase 2 studies run to test the concept, you know, with that repurposing drug, you know, if you don't have the IP protection, then who's going to fund the phase 3? Right, because it's not the government, it's not an academic lab, it's not an institute, so it's got to be a commercial partner, right? And so if they don't see that it's got any IP protection, I mean, biotechs sort of view this somewhat differently from big pharma, but you know, or you might have a backup molecule that you think, well, actually we can, you know, file a patent on that, and there's enough differentiation from the backup from the lead molecule. But you can't be, I suppose, to put it bluntly, naive about the IP. Yeah, you've got to have a novel position on how you're differentiating from what's already out there. So I think that's, again, what's different about this group and this grant board, that you've got to think maybe a bit more like the pharmaceutical industry, and thinking how your research is going to translate into treatment.
Ooh, you're on mute, Jess.
Yeah.
It was going to happen to somebody, so it happened to me. So thank you. I guess before we wrap up for the Q&A session, are there any kind of final thoughts from both of you about what you're looking to see in applications for this round?
So one thing occurred to me was looking through the guiding principles, and we're talking about, you know, as Jill rightly said, quite often academics can have the great ideas, the great biology, understanding disease pathophysiology, but maybe don't have the skill set to do all of the things that we've talked about and take it forward. But actually those skill sets are out there, So, you know, things like pharmacokinetic studies, let's say, you know, there are CROs out there who can deliver those for not unreasonable costs, especially in early stages if you're not necessarily doing everything to what's called GLP. And they can, so CROs are a good source, other collaborators. So have a look at those kind of guiding principles. And if you think there's an area where you would be weak, think about who can I collaborate with? And there may be academic collaborators. There are certainly some good academic centers that do this type of research all the time, but also, you know, potentially in industry collaborators that you might have contacts with might be able to assist or kind of come on board. So just think about where can you get that skill set. So it's really the whole, you know, a real team effort, drug discovery. I think that's one of the things, or new therapeutic discovery. It's a real team effort. And I think that's one of the really exciting things, is to bring all of these different disciplines together to bear on that, you know, tough challenge.
That's a great point, Richard. And we're happy to fund subcontracting studies to CROs. So they are covered under eligible costs within this grant round. And Jill?
Yeah, I'm just checking I'm not on me. Yeah. So I think for me, you know, it is an ecosystem. You know, it's not just about industry or academia or biotech. All three need to work together. And I think, you know, where you can have collaborations with, you know, people that have a different area of expertise, you know, I think that does help with a more holistic approach. So, you know, I think we rely on the academics to really come up with some brilliant targets. You know, that's not the strength, particularly of the pharmaceutical industry, but where we can enable those targets to become drugs down the line. I think that's where the partnership comes in. So, you know, it's not that we aren't identifying new targets, but I think, you know, in terms of, you know, having a different aspect, different view and different approach, different expertise, you know, that's really important that we have a partnership there. And so, you know, yeah, I think working together and just, you know, really focusing. I think for me, you know, the most successful programs internally is where you have a really good focus and really realistic about the deliverables, you know. So with grant applications, you know, sometimes you see very overly ambitious grants where you've got 5 or 6 different goals and maybe aren't able to deliver on all of them very well. Whereas if you concentrate on 2 or 3 that really are sort of incremental steps to changing the way we think about that target, so that maybe a follow-up grant will elaborate on that. So, you know, it's all about being realistic, having clear milestones, and just focus on the deliverables.
Exactly. Thank you very much, both of you. We're going to bring Ange back as well now for this final Q&A session. And we have one question in the chat, which is probably for Richard and Jill, which is what level of target novelty is preferred?
It's a good question. So, and it might not necessarily be the target itself that's novel, but there needs to be, I think, there does need to be some element of your approach being novel, let's say. So let's say the target is known, but you've identified new biology, which suggests that targeting in this particular way is actually more relevant or is actually going to make a difference. So just going back to the commercial side of things, you know, the kind of the commercial pathway forward that Jill just talked about, if you're doing the same thing that everyone else is doing, then obviously that's going to negatively impact on your application, because the foundation wants to be funding things that other people aren't doing. So I think that's the basic cutoff, is have you got something new, or are you aware that other people are actually already doing it and maybe are ahead of you? So that has been a consideration in some of our discussions, where someone's come with a proposal that's a strong proposal, but we're aware, or that it's in the public domain, that other approaches targeting the same pathway, or say exactly the same approach or very similar approach, are much further ahead. So that's, I hope that answers your question.
Yeah, and I think we've seen examples of that with some of the ASO approaches, right, where there are groups and companies and biotechs, you know, way ahead of some of the ideas that we've heard about. And so, you know, just think, well, you know, on paper, does this realistically look like you can differentiate? And if it doesn't, then you've got to rethink really and think about another approach, another modality, you know, another way of targeting the same protein, but not go with something that you're, you know. And again, in drug discovery companies, you know, if a company is like 3 or 4 years ahead of you, you've got to have a damn good argument of why you're going to launch a program, right? And you'll get pushback on that internally. And I think it should be the same with these grant applications.
Perfect, thank you. And Giacomo, I think you had a few questions that were submitted beforehand, is that right?
Yes, one for Ange. So Ange, in your opinion, what are the key elements that make a research proposal stand out?
Oh, that's a really good one. I think it would just be something that I could read really easily and would have a tangible outcome that I could really resonate with, that you could really see where that research is going and get excited by it.
That's great to hear. I think there is one question for Richard and Jill that has not been touched. I feel like it's about what elements or themes in proposal that tended to receive higher evaluation from the committee in the past. What type of themes and elements do you think are valued?
Yeah, I don't know. I mean, the thing is, you know, a good proposal when you read it, but you know, everything that we've talked about, if you're ticking all of those boxes and you're presenting it in a way that's what, you know, we've not. I mean, Anne just talked about this, but actually, you know, the clarity of how you present and how you write your application can make a really big difference. Not being too broad in your objectives, as Jill has mentioned, you know, having multiple different objectives, work streams, being so, you know, focused, well-written applications that address the kind of key points that we've already discussed, I think are the ones that tend to stand out.
Yeah, I think focus and clear objectives, well-written grant usually means well-executed science in my experience. So, you know, obviously novelty, innovation, but also a fair and transparent critique of the literature. So sometimes when you go out and follow up on, you know, a citation, you know, one side of the story is being presented and it's not particularly balanced. So, you know, and actually having very accurate proposals as well, because, you know, it's like anything in life, you know, if it doesn't look like things have been done properly, or, you know, even sometimes you get a lot of mistakes in some grants as well, you know, where you think, well, how much attention to detail are people giving? So having really professional, accurate, you know, clear deliverables and so on, I think is very important from my perspective. And that's true of any grant board, not just this one.
Okay.
Thank you so much. I'm mindful of the time, so I want to thank everyone who attended, our speakers. And I want to remind everyone that the deadline is midday on the 17th of May, and you will be able to see this recording on YouTube very soon. Thank you all.
Thank you.
Thank you.
Thanks all.